What is glycogenolysis? Glycogenolysis is the metabolic process of breaking down glycogen, which is the stored form of glucose, into individual glucose molecules. This process involves the systematic breakdown of glycogen, a large branched polymer of glucose, into its constituent glucose units. Glycogenolysis is essential for several important physiological functions. It provides energy during fasting periods when glucose is not being consumed through diet. It helps maintain stable blood glucose levels, which is crucial for brain function and overall homeostasis. And it responds to increased energy demands, such as during exercise or stress. Glycogenolysis is a fundamental process in human metabolism that helps ensure a steady supply of glucose, the body's primary energy source. Glycogenolysis, the breakdown of glycogen into glucose, occurs primarily in two main locations in the body. The most significant sites are the liver and skeletal muscles, with minimal activity in other tissues like the kidneys and adipose tissue. In the liver, glycogenolysis serves a critical function of maintaining blood glucose levels. The liver breaks down its glycogen stores into glucose, which is then released into the bloodstream. In skeletal muscles, glycogenolysis has a different purpose. Rather than releasing glucose into the bloodstream, muscle cells break down glycogen to provide energy locally for muscle contraction. At the cellular level, glycogenolysis takes place in the cytoplasm of cells, not within any specific organelle. The enzymes responsible for breaking down glycogen operate in the cytosolic compartment, where glycogen is stored as granules. Let's compare the key differences between glycogenolysis in the liver and skeletal muscles. The liver breaks down glycogen primarily to maintain blood glucose levels, releasing free glucose into the bloodstream. Muscle cells, in contrast, use glycogenolysis to provide energy locally for muscle contraction, without contributing to blood glucose levels. The liver produces glucose that can leave the cell and enter the bloodstream, while muscles produce glucose 6-phosphate that must be used within the muscle cell. Liver glycogenolysis is primarily regulated by hormones like glucagon and insulin, whereas muscle glycogenolysis responds mainly to allosteric factors like AMP and calcium. The liver is most active during fasting periods between meals, while muscle glycogenolysis increases during exercise and intense physical activity. To summarize the key points about where glycogenolysis occurs in the body, glycogenolysis primarily occurs in the liver and skeletal muscles, taking place within the cytoplasm of cells. The liver releases glucose into the bloodstream to maintain glucose levels, while muscles use the energy locally for contraction. There is also minor glycogenolysis activity in other tissues, such as the kidneys and adipose tissue. Glycogen phosphorylase is the principal enzyme responsible for initiating glycogen breakdown during glycogenolysis. Glycogen phosphorylase exists as a dimer, consisting of two identical protein subunits that work together for its enzymatic function. The enzyme requires pyridoxal phosphate, a derivative of vitamin B6, as a cofactor for its catalytic activity. Glycogen phosphorylase specifically cleaves alpha-1,4 glycosidic bonds in the glycogen chain. When the enzyme acts on glycogen, it breaks the alpha-1,4 bonds and releases glucose 1-phosphate units. Each cleavage reaction releases a single glucose 1-phosphate unit, which can then enter metabolic pathways. To summarize, glycogen phosphorylase is the principal enzyme in glycogenolysis, with a dimeric structure requiring vitamin B6 as a cofactor and releasing glucose 1-phosphate units through its action. Glycogen phosphorylase catalyzes the breakdown of glycogen into glucose 1-phosphate through a process called phosphorylasis. Unlike hydrolysis, which uses water, phosphorylasis uses inorganic phosphate to break glycosidic bonds. Glycogen phosphorylase works specifically from the non-reducing ends of glycogen chains. The enzyme attaches to the non-reducing end of a glycogen chain, and with the help of inorganic phosphate, it cleaves off one glucose unit at a time. This process repeats sequentially, removing one glucose unit after another, generating glucose 1-phosphate with each cleavage. Importantly, glycogen phosphorylase can only continue cleaving until it reaches about 4 glucose units from a branch point. At this point, another enzyme, the debranching enzyme, is required to continue the process. To summarize, glycogen phosphorylase works by phosphorylasis to sequentially remove glucose units from the non-reducing ends of glycogen, producing glucose 1-phosphate until it reaches about 4 glucose units from a branch point. Phosphoglucomutase is a crucial enzyme in the glycogenolysis pathway. This enzyme catalyzes an important isomerization reaction, converting glucose 1-phosphate to glucose 6-phosphate. The enzyme catalyzes the conversion between two isomers of glucose phosphate. This reaction is an isomerization, where the phosphate group moves from carbon 1 to carbon 6. The mechanism involves a temporary addition of another phosphate group, forming glucose 1,6-bisphosphate as an intermediate. This conversion is crucial because glucose 6-phosphate is a versatile metabolic intermediate that can enter various pathways. In the liver, glucose 6-phosphate can be converted to free glucose by the enzyme glucose 6-phosphatase, allowing glucose to be released into the bloodstream to maintain blood glucose levels. In muscle tissue, which lacks glucose 6-phosphatase, glucose 6-phosphate enters glycolysis directly to produce energy in the form of ATP. In summary, phosphoglucomutase provides metabolic flexibility by converting glucose 1-phosphate from glycogen breakdown to glucose 6-phosphate, which can then feed into multiple metabolic pathways depending on the tissue and the body's needs. Glucagon is a hormone secreted by pancreatic alpha cells when blood glucose levels are low. When blood glucose drops, alpha cells release glucagon into the bloodstream. Glucagon primarily targets the liver, where it binds to specific receptors on the cell membrane. When glucagon binds to its receptor, it triggers a signaling cascade inside the liver cell. The activated receptor stimulates an enzyme called adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP or cAMP, which acts as a second messenger. Increased cAMP activates protein kinase A, an enzyme that adds phosphate groups to other proteins. Protein kinase A then activates phosphorylase kinase through phosphorylation. Finally, phosphorylase kinase activates glycogen phosphorylase, the key enzyme that catalyzes glycogenolysis. Activated glycogen phosphorylase breaks down glycogen into glucose 1-phosphate molecules, which can then be converted to glucose 6-phosphate and either used by the cell or released into the bloodstream. This entire cascade ensures that when blood glucose levels are low, the liver breaks down its glycogen stores to release glucose and restore normal blood glucose levels. Epinephrine, also known as adrenaline, is a hormone released during the fight or flight response. When released, epinephrine stimulates glycogenolysis in both liver and muscle tissues to rapidly provide glucose for energy. In the liver, epinephrine works through a cAMP-dependent pathway, similar to glucagon. It binds to beta-adrenergic receptors, activating G proteins and adenylyl cyclase. This increases CAMP levels, which activates protein kinase A. PKA then phosphorylates and activates glycogen phosphorylase, initiating glycogenolysis. In muscle tissue, epinephrine operates through two mechanisms. First, the CAMP pathway via beta-adrenergic receptors, similar to the liver. But muscle tissue also responds through alpha-adrenergic receptors, activating a calcium-dependent pathway. This involves phospholipase C and IP3 production. The IP3 triggers calcium release from the sarcoplasmic reticulum. This calcium then activates phosphorylase kinase, which in turn activates glycogen phosphorylase, leading to glycogenolysis. Both pathways lead to increased glycogenolysis, releasing glucose 1-phosphate that can be converted to glucose 6-phosphate. In the liver, this can become free glucose to raise blood glucose levels. To summarize, epinephrine stimulates glycogenolysis in both liver and muscle through CAMP-dependent pathways, with muscle having an additional calcium-dependent mechanism, allowing for a rapid response during stress or physical activity. Glycogen phosphorylase activity is finely controlled through allosteric regulation. Glycogen phosphorylase has an active site where catalysis occurs and separate allosteric sites where regulatory molecules can bind. When cellular energy is low, AM levels increase. AMP is an allosteric activator of glycogen phosphorylase. When AMP binds to the allosteric site, it causes a conformational change that activates the enzyme. The activated enzyme can now more efficiently break down glycogen, releasing glucose 1-phosphate molecules. In contrast, when energy is abundant, both glucose and ATP act as allosteric inhibitors of glycogen phosphorylase. When glucose levels are high, it binds to the allosteric site and inhibits the enzyme. Similarly, high ATP levels indicate that the cell has sufficient energy. ATP binds to another allosteric site, further stabilizing the inactive conformation. With both glucose and ATP bound, the enzyme adopts a fully inhibited conformation, significantly reducing glycogenolysis. These regulatory mechanisms involve shifting the enzyme between two conformational states, the tense T state with lower activity and the relaxed R state with higher activity. In summary, AMP shifts the enzyme toward the active R state, while glucose and ATP promote the inactive T state. This allows glycogen breakdown to be precisely regulated based on the cell's energy status. We'll now explore how glycogen phosphorylase is regulated through phosphorylation. Glycogen phosphorylase exists in two main forms, the less active phosphorylase B and the more active phosphorylase A. The conversion from phosphorylase B to phosphorylase A is catalyzed by an enzyme called phosphorylase kinase. Phosphorylase kinase adds a phosphate group to phosphorylase B, converting it to phosphorylase A. This phosphorylation dramatically increases the enzyme's activity. Phosphorylase A is much more efficient at breaking down glycogen than phosphorylase B. This phosphorylation-based regulation is initiated by hormonal signals like glucagon and epinephrine. When these hormones bind to cell receptors, they trigger a signaling cascade. This cascade activates several enzymes in sequence, ultimately leading to the activation of phosphorylase kinase. This phosphorylation-based regulation system provides rapid and sensitive control of glycogen breakdown in response to hormonal signals. The system allows for rapid responses to metabolic demands, amplification of hormonal signals, and precise control of glycogen breakdown when energy is needed. Calcium ions serve as critical second messengers in the regulation of glycogenolysis, particularly in muscle tissue during exercise. When muscles contract, calcium is released from the sarcoplasmic reticulum into the sarcoplasm. This sudden increase in cytosolic calcium concentration triggers a coordinated response that links muscle contraction with energy production. Calcium directly binds to phosphorylase kinase, which has a specific calcium binding subunit, triggering its activation. The activated phosphorylase kinase then phosphorylates and activates glycogen phosphorylase, the key enzyme in glycogenolysis. Activated glycogen phosphorylase begins breaking down glycogen into glucose 1-phosphate molecules, providing energy for the contracting muscle. This calcium-mediated mechanism ensures that glycogen breakdown is tightly coordinated with muscle contraction, providing energy precisely when and where it is needed during exercise. This calcium-dependent regulation is rapid, operating within milliseconds, and functions independently of hormonal signals. It provides localized energy at the site of muscle contraction without causing systemic changes in blood glucose levels. Liver glycogenolysis is a critical process that maintains blood glucose levels between meals. When blood glucose begins to fall after a meal, the liver breaks down glycogen stores to release glucose. This released glucose enters the bloodstream, maintaining blood glucose levels and preventing hypoglycemia. Glycogenolysis is one of the body's first responses to declining blood glucose, occurring within a few hours after a meal and before gluconeogenesis becomes active. Without glycogenolysis, blood glucose levels would rapidly fall below the normal range, leading to hypoglycemia during fasting periods. Glycogenolysis is essential for preventing hypoglycemia during fasting periods. It provides a rapid source of glucose before other metabolic pathways like gluconeogenesis become fully active. As one of the body's primary metabolic defenses, liver glycogenolysis ensures a constant supply of glucose to the brain and other vital organs between meals. During exercise, muscle cells need a rapid source of energy. Muscle glycogenolysis is crucial for providing immediate energy, especially during high-intensity activities. During exercise, glycogen in muscles is broken down through glycogenolysis, releasing glucose 6 phosphate. Glucose 6 phosphate enters glycolysis, a metabolic pathway that generates ATP rapidly. This process is especially important because it can generate energy without requiring oxygen, making it crucial for intense bursts of activity like sprinting or weightlifting. This makes muscle glycogenolysis critical for short-duration, high-intensity exercise where ATP demands are high and oxygen utilization cannot keep pace. Glycogenolysis has important functions beyond simply generating energy. The glucose 6-phosphate released from glycogen can support various other cellular activities. One crucial pathway is the pentose phosphate pathway, which uses glucose 6-phosphate to generate NADPH. NADPH is essential for biosynthetic reactions like lipid synthesis and provides electrons for antioxidant defense systems. Glucose from glycogenolysis also provides substrates for protein glycosylation, a critical post-translational modification. Glycosylation is essential for cell-cell recognition, proper protein folding, and protein stability. In the liver, glycogenolysis plays a particularly important role in supplying glucose for various tissues and cellular processes. For example, red blood cells rely on the pentose phosphate pathway for antioxidant defense. The brain needs glucose for proper functioning, and immune cells require glycosylation for cell recognition and signaling. Glycogen storage diseases, or GSDs, are a group of inherited disorders that affect enzymes involved in glycogen metabolism. In this section, we'll focus specifically on disorders that affect glycogenolysis, the breakdown of glycogen. McArdle disease, also known as glycogen storage disease type 5, is caused by a deficiency in muscle phosphorylase. In this disorder, the enzyme that breaks down glycogen in muscle tissue is missing or dysfunctional. This prevents muscles from accessing stored glycogen during exercise. Patients with McArdle disease typically experience exercise intolerance, muscle cramps, and pain when physically active. They may also experience the second wind phenomenon, where symptoms temporarily improve after rest or continued mild exercise. HERS disease, or glycogen storage disease type 6, is caused by a deficiency in liver phosphorylase. In this disorder, the enzyme that breaks down glycogen in liver tissue is missing. This prevents the liver from releasing glucose into the bloodstream when blood sugar levels drop. Patients with HERS disease typically experience hypoglycemia, or low blood sugar, especially during fasting. They may also have an enlarged liver and growth retardation. However, the symptoms are generally milder than in other glycogen storage diseases. Let's compare McArdle and HERS diseases. Both involve deficiencies in phosphorylase enzymes, but they affect different tissues with distinct clinical presentations. McArdle disease affects muscle phosphorylase, causing exercise intolerance in skeletal muscles. It typically becomes apparent in childhood or early adulthood. HERS disease affects liver phosphorylase, leading to hypoglycemia. It generally manifests in early childhood and affects the liver's ability to maintain blood glucose levels. It's important to note that both McArdle and HERS diseases are inherited in an autosomal recessive pattern, meaning both parents must carry a copy of the mutated gene for a child to be affected. Von Gierke disease is a classic glycogen storage disorder that reveals important insights about glycogenolysis. Also known as glycogen storage disease type 1, this rare autosomal recessive disorder affects approximately 1 in 100,000 births. The fundamental defect is a deficiency of glucose 6-phosphatase, a critical enzyme that converts glucose 6-phosphate to free glucose in the liver. In this disorder, glycogenolysis proceeds normally, breaking down glycogen to glucose 6-phosphate. However, the critical final step is blocked. Due to glucose 6-phosphatase deficiency, glucose 6-phosphate cannot be converted to free glucose, creating a metabolic bottleneck. This biochemical block leads to several severe clinical manifestations. The liver is particularly affected, becoming massively enlarged due to glycogen and fat accumulation. Let's understand the mechanism. Since glucose 6-phosphate cannot be converted to glucose, the liver cannot release glucose into the bloodstream during fasting. This leads to severe hypoglycemia, as the body cannot maintain blood glucose levels during fasting periods, even though glycogen is abundant in the liver. Secondary metabolic consequences include lactic acidosis from increased glycolysis, hyperuricemia, and hyperlipidemia. Von Gierke disease illustrates how a single enzyme deficiency in the glycogenolysis pathway can disrupt multiple metabolic processes throughout the body. Diagnosing glycogenolysis disorders requires a comprehensive approach using multiple testing methods. Enzyme assays from tissue biopsies are a cornerstone of diagnosis. Samples are usually taken from liver or muscle tissue to directly measure the activity of enzymes involved in glycogen breakdown. Genetic testing identifies mutations in genes responsible for glycogenolysis. This allows precise diagnosis of specific disorders like von Gierke disease, Pompe disease, and others. It also helps identify carriers and enables genetic counseling. Exercise tolerance tests examine how the body responds to physical activity. In glycogenolysis disorders, patients often show abnormal patterns of lactate production, glucose utilization, and fatigue. These tests are particularly valuable for diagnosing muscle-related glycogen storage diseases. Imaging studies provide non-invasive visualization of affected organs. Ultrasound, MRI, CT scans, and fibroscan can detect hepatomegaly or enlarged liver, a common feature in many glycogenolysis disorders. These scans can also identify muscle abnormalities and help monitor disease progression. By combining different diagnostic approaches, clinicians can differentiate between various glycogenolysis disorders. Each disorder has a unique pattern of enzyme deficiencies, genetic mutations, and clinical features. This comprehensive approach ensures accurate diagnosis, which is essential for proper management and treatment. Treatment strategies for glycogenolysis disorders focus on managing symptoms and preventing metabolic crises. Dietary management is the cornerstone of treatment. Patients need frequent meals every 2 to 3 hours to maintain stable blood glucose levels. A diet high in complex carbohydrates provides sustained energy release. Protein supplementation may be necessary depending on the specific disorder. Strictly avoiding fasting periods is critical, as even short periods without food can trigger dangerous hypoglycemia. Cornstarch therapy is a cornerstone treatment for many glycogenolysis disorders, particularly those affecting the liver, like von Gierke disease. Uncooked cornstarch works as a slow-release form of glucose, typically dosed at 1 to 2 grams per kilogram body weight every 4 to 6 hours. This is especially important overnight when the fasting period is longest. The slow digestion of cornstarch provides a steady glucose release, preventing dangerous hypoglycemia. Newer modified starch products with longer duration of action have been developed, allowing for less frequent dosing and better overnight coverage. While dietary management and cornstarch therapy are the current standards of care, several emerging therapies show promise for treating glycogenolysis disorders. Enzyme replacement therapy uses recombinant enzymes delivered intravenously to compensate for the specific enzyme deficiency. Gene therapy represents a potentially permanent solution. Using viral vectors to deliver functioning copies of the affected genes, it could correct the underlying genetic defect. Additional supporting therapies are often necessary to manage specific complications of glycogenolysis disorders. Growth hormone may be used to improve growth and metabolic control in patients with growth delays. For severe liver disease, liver transplantation can be considered. Allopurinol helps control elevated uric acid levels often seen in these disorders. ACE inhibitors may be used to manage kidney complications that can develop over time. In summary, treatment strategies for glycogenolysis disorders combine dietary management, cornstarch therapy, and supporting medications with promising emerging options on the horizon. Treatment must be individualized based on the specific disorder, age of the patient, and severity of symptoms. Regular monitoring by a specialized metabolic team is essential. Glycogenolysis is the enzymatic process of breaking down glycogen into glucose 1 phosphate, which is converted to glucose for energy use. Glycogenolysis occurs primarily in the liver and skeletal muscle, but through slightly different pathways. Both tissues use glycogen phosphorylase as the rate-limiting enzyme, followed by debranching enzyme and phosphoglucomutase. The critical difference is that liver cells possess glucose 6-phosphatase, allowing them to release free glucose into the bloodstream to maintain blood glucose levels. Muscle cells lack this enzyme and use glucose primarily for their own energy needs. Glycogenolysis is tightly regulated through multiple mechanisms. Hormonal control includes glucagon and epinephrine, which promote the process, while insulin inhibits it. Allosteric regulators include AMP, which activates an ATP, and glucose 6-phosphate, which inhibit the process. Enzyme phosphorylation through protein kinase A and phosphorylase kinase plays a crucial role. In muscle, calcium release during contraction directly activates phosphorylase kinase, rapidly mobilizing glycogen during exercise. Glycogenolysis serves three primary physiological functions. First, it maintains blood glucose levels, especially during fasting periods, which is essential for brain function. Second, it provides rapid energy during exercise, particularly during high-intensity activities when oxygen delivery isn't sufficient for aerobic metabolism. Third, it supports the body's immediate energy needs during stress responses and helps cells adapt to changing metabolic demands. The clinical significance of glycogenolysis is most evident in glycogen storage diseases, where genetic enzyme deficiencies disrupt the normal breakdown of glycogen. Type 1 GSD, or von Gierke disease, involves glucose 6-phosphatase deficiency, preventing liver glucose release. This causes severe hypoglycemia, hepatomegaly, and growth retardation. Type V GSD, or McArdle disease, involves muscle glycogen phosphorylase deficiency, causing exercise intolerance and muscle cramps because muscle cells cannot use their glycogen stores during activity. Understanding glycogenolysis has significant implications for medical practice. It's crucial for diabetes management, where disrupted glycogen metabolism contributes to glucose dysregulation. It's essential knowledge for exercise physiology, sports medicine, and critical care, where stress can induce hyperglycemia through accelerated glycogenolysis. It informs genetic counseling for glycogen storage diseases and enables drug development targeting specific steps in the pathway. In conclusion, glycogenolysis represents a fundamental metabolic process that maintains energy homeostasis and ensures glucose availability when needed, highlighting the elegant balance of our metabolic systems.